In the Indus Valley, in the year 2000 BC, while the Egyptian New Kingdom was still 4 centuries from its founding, a civilization we have only partially decoded was cataloging the properties of a single tree with the systematic precision of a pharmaceutical laboratory. They called it Nimba. Later Sanskrit texts would call it Sarva Roga Nivarini, the cure of all ailments. But it was not only healers who prized it. Farmers pressed its seeds into oil, ground its bark into powder, and buried its crushed leaves in the soil around their crops. Not because a priest told them to, because generation after generation, they had watched what happened to crops protected by this tree, and what happened to the insects that encountered it. The Charaka Samhita, one of the foundational texts of Ayurvedic medicine, composed between 900 and 600 BC, and encoding knowledge far older, dedicates extensive passage to the Neem tree as a Tikta Skandha, a bitter group plant, documenting its capacity to disrupt the life cycles of parasites, fungi, and destructive insects without harming the farmer, the soil, or the animals grazing nearby. This was not superstition recorded in an ancient book. This was field-verified, generationally tested applied entomology, practiced across an agricultural civilization spanning over 1 million square kilometers, feeding populations whose infrastructure rivaled anything Rome would build 2 millennia later. The civilization collapsed. The records were buried. The tree remained. And for the next 4,000 years, it stood in the fields of South Asia, still doing its work, still protecting crops, still poisoning nothing, while the world around it forgot what it was. In 2022, the global chemical pesticide market was valued at approximately $84 billion. $84 billion every year to solve a problem that a farmer in the Indus Valley solved 4,000 years ago with a tree that grows freely across an entire subcontinent and requires no synthesis, no patent, and no annual renewal. The architecture of the modern chemical pesticide industry is not built on efficacy. It is built on dependency engineering. A product that permanently solves an insect problem is a product that sells once. A product that temporarily suppresses an insect population while simultaneously selecting for genetic resistance in the survivors, that is a product that sells indefinitely. This is not a theory. It is a documented, peer-reviewed biological phenomenon called pesticide resistance, and it is the industry's most reliable growth engine. Here is how the cycle operates. A synthetic pesticide, organophosphate, pyrethroid, neonicotinoid, is applied to a crop. It kills the susceptible majority of the target insect population. But in any population, there exist individuals carrying random genetic mutations that confer partial resistance to the compound's mechanism of action. Those individuals survive. They reproduce within 3 to 7 generations, which, for an insect with a 2-week life cycle, can represent a single growing season. The resistant genotype dominates the local population. The pesticide is now largely ineffective. The farmer, watching his crop fail, does not question the system. He is told he needs a higher concentration, a different compound, or a combination product. He buys it. The cycle begins again. The chemical companies are not unaware of this mechanism. Their own internal research documents it. The industry term for it is resistance management, a phrase that frames the biological arms race as a logistical challenge to be administered rather than a fundamental indictment of the approach. The recommended solution to resistance is rotation, cycling between different classes of pesticides on a schedule. This is sold as responsible farming. What it actually does is systematically expose insect populations to every available chemical class in sequence, accelerating the development of multi-compound resistance across all of them simultaneously. Meanwhile, the compound that the Indus Valley farmers understood in 2000 BC, azadirachtin, the primary active molecule in Neem, does not trigger resistance. It has been in agricultural use, in one form or another, for 4 millennia, and there is not a single peer-reviewed study documenting a field population of any insect species developing resistance to it. not one. The mechanism that makes azadirachtin effective is so fundamentally different from synthetic pesticides that resistance to it would require an insect to rewire the hormonal architecture of its own developmental biology, a mutation so metabolically expensive that natural selection has never produced it in recorded agricultural history. The industry knows this. The industry has known this since azadirachtin was first formally isolated and characterized by chemist Heinrich Schmutterer in 1968. It has known this since the European Patent Office's landmark 1995 ruling on the Neem patent controversy, a case in which the Indian government and international environmental coalitions successfully challenged a U.S. patent attempt on Neem-based fungicide, establishing that this knowledge had been in the public domain for thousands of years and could not be privately owned. It knows. It simply has no profit motive to tell you. To understand why azadirachtin is categorically different from every synthetic pesticide ever manufactured, you need to understand the difference between killing and disrupting. Every major class of synthetic pesticide operates on a kill mechanism. Organophosphates inhibit acetylcholinesterase, an enzyme essential for nerve signal termination, causing the target insect's nervous system to fire continuously until muscular failure and death. Pyrethroids force sodium-ion channels in nerve cell membranes to remain permanently open, producing the same outcome. Neonicotinoids bind irreversibly to nicotinic acetylcholine receptors in the central nervous system, producing hyperexcitation and death. These are blunt instruments, effective, fast, and, crucially, non-selective. The same neural mechanisms they destroy in a caterpillar exist, in homologous form in a honeybee, a ground beetle, a parasitic wasp, and at low enough concentrations in human neural tissue. Azadirachtin operates on an entirely different principle. It does not kill, it confuses. The molecule's primary target is not the insect nervous system. It is the insect endocrine system, specifically the hormonal cascade that governs metamorphosis, reproduction, and appetite. Here is the mechanism precisely. Insects develop through a process governed by two hormones held in careful balance: ecdysone, which triggers molting and developmental transitions, and juvenile hormone, which suppresses premature metamorphosis. The timing and ratio of these two hormones determine when a larva molts, when it pupates, and when it emerges as a reproductive adult. The entire developmental life cycle of every insect on Earth is orchestrated by this hormonal dialogue. Azadirachtin is a liminoid triterpene, a large, structurally complex molecule that bears a close geometric resemblance to ecdysone. When an insect ingests plant tissue containing azadirachtin, the molecule enters the hemolymph, insect blood, and reaches the prothoracic gland, the organ responsible for producing ecdysone. There, it acts as a hormone mimic and disruptor simultaneously. It binds to ecdysone receptor sites with sufficient affinity to block the natural hormone signal, but insufficient activity to trigger the developmental response. The result is a hormonal traffic jam. The insect cannot complete its molt. It is arrested mid-development, unable to transition from one larval stage to the next, unable to pupate, unable to reach reproductive maturity. Feeding behavior is also directly suppressed through a secondary mechanism. Azadirachtin disrupts the chemoreceptor neurons in the insect's maxillary palps, the sensory organs used to evaluate food palatability, producing a powerful anti-feeding effect.